Good evening, Tarun. Good evening, Vanshika. Okay, so I'm Rahul Saha, and last time we had talked about the incompatibility issues, the heteromorphic system, okay, where the incompatibility was due to the morphological changes, right? The morphological flaws or the morphological dissimilarities. Then we have talked about the pin flowers, which bear the long styles and short stamens, while the thumb flowers, which have short style and long stamens. Then we have talked about the specific homomorphic system where the incompatibility is not connected with morphological differences whatsoever, right? So it could be again sporophytic and gametophytic. In sporophytic, we have talked about that the incompatibility is governed by a single gene, like capital S, where S is dominant in multiple allelism, and then you can say the compatibility reaction of pollen is controlled by the genotype of the plant on which the pollen is produced and not by the genotype of the pollen itself. And in gametophytic control, we had learned that the incompatibility of the pollen is controlled by its own genotype and not by the genotype of the plant on which it is produced, right? So now we'll talk about the next thing known as the biological significance significance of self incompatibility. So self incompatibility may be due to a we know that pollen stigma interaction interaction or pollen tube style interaction. These are the two factors governing the basics of self incompatibility. One more you can add if you want deep knowledge, like pollen tube plus ovule interaction. Pollen tube plus ovule interaction. Now, in self incompatible fruit trees, it is necessary to plant two cross compatible varieties to ensure fruitfulness. Now, self compatibility means it can be used in hybrid seed production, right? The application could be that self compatibility or self incompatibility, I should be saying incompatibility can be used to produce, you can say, hybrid seed production or to produce hybrid seeds. Then, in many cases, the self fertile forms are highly desirable, and in such case, it would be extremely useful to again go for elimination of self incompatibility. So in nature, what happens? There are something known as inbreeding and outbreeding. Okay, then inbreeding leads to inbreeding, depression. So we have inbreeding and outbreeding of the plants, which are again regulated by inbreeding and outbreeding. They are again regulated by, you can say, intraspecific and interspecific incompatibility. Intraspecific and interspecific incompatibility. Okay. Now, extensive selfing, if you want to go for, suppose a scientist wants to go for extensive selfing, so he is going for selfing, selfing, and selfing incessantly. So, extensive selfing of plants can lead to production of highly homozygous individuals. Okay, again, which will lead to what? We have talked about this phenomenon, which will lead to again inbreeding depression, which will have a very low fertility and a low, you can say, survivability probability. Then you can say one more thing is as far as competitive examinations are concerned, so extensive selfing will again lead to inbreeding depression. Inbreeding depression. Now, nature has put natural selection. Natural selection means in the way of or in the form of self incompatibility. So, nature has put self incompatibility as a measure to check this phenomenon so that inbreeding or inbreeding depression can be, you can say, circumvented, can be avoided. That is one way the nature can take care of itself, because I told you in the ecosystem also nature is pretty selfish. It doesn't allow you to reach the carrying capacity k. It doesn't allow you to reach the full biotic potential 1 minus n by k, right? So you cannot realize the full biotic potential, you cannot reach the carrying capacity, else the resources will become completely used up and the future generations would be deprived of the resources, and thereby you will see a sharp decline, a plummet in the number of species due to starvation, like what we saw in the J curve, right? Geometric curve. You go up and up in case of reindeer, and when the eventual resources are over, eventually the resources are over, then they see or they encounter a sharp decline, right? A steep decline, you can say. So you can say nature has imposed self-incompatibility in order to check inbreeding depression. On the other hand, interspecific incompatibility, it brings about what? Inter, just think from the evolution perspective. Specific incompatibility. From evolution perspective, it brings about what? It brings about reproductive isolation or not. This is a prime exemplar of reproductive isolation. Okay. Now we had learnt about various kinds of speciation. So is it not allopatric speciation? We had learnt about aeropatric, parapatric, sympatric, peripatric. So is it not allopatric speciation, right? And we'll need to development of or emergence of new races or new species, I can say, right? Now, this can happen due to the process of mutation and open hybridization. So these are the tricks by which nature can generate. Good evening, Ayman. So these are the tricks by which nature can generate new species in the long run. Okay. Now we'll come to the next topic in this chapter known as parthenogenesis. Parthenogenesis. In parthenogenesis, what happens is, I'll first write the definition for you. Parthenogenesis is the formation of embryo directly from egg cell or a male gamete. Or male gamete. Now, this is the meaning of parthenogenesis. Whereas one more thing is asked in board examination, that is known as apogamy. Now, parthenogenesis, we know embryo is directly from egg cell or it can be produced from a male gamete, whereas apogamy means development of a sporophyte, that is embryo, out of any gametophytic cell without fusion of gametes. That's the difference. That means it is the development of you can say a sporophyte that is embryo out of any gametophytic cell without the fusion of gametes. Without fusion of gametes. That is known as apogamy. Now, one more thing I'll tell you, something known as apospory. Okay, parthenogenesis, then apogamy, then something is known as apospory is also asked in the board examination. We'll talk about apospory. In case of apospory, it represents the formation of, you can say it represents the formation of what? The formation of gametophyte. Gametophyte means the pollen or embryo sac from a sporophyte without undergoing meiosis. It's a very, very special event. So it represents, you can say, the formation of gametophyte that is pollen or embryo sac slash embryo sac from a sporophyte here, not from gametophyte, from a sporophyte without even undergoing meiosis. Without undergoing meiosis event. So what you can say, parthenogenesis is what? Parthenogenesis does. I can say as per the definitions if I compare. So parthenogenesis is the formation of embryo directly from egg or a male gamete, right? So without fusion of gametes, part of apogamy does fit into parthenogenesis, right? So parthenogenesis does can be called as a type of apogamy or not, right? So that's a pretty good neat question. So parthenogenesis is a type of apogamy. Okay, so parthenogenesis is a type of apogamy. So in plants like Solanum nigrum, okay, Solanum nigrum, the development of haploid egg into embryo or into an embryo has been observed by scientists and has been documented too, right? It has been observed. Now this leads to formation of haploid embryo and plant as well. This leads to formation of haploid embryo and plant as well. Now this is a special type because only haploid embryo has been produced. Now this type of parthenogenesis is thus referred to as haploid parthenogenesis. A new term. This is referred to as a haploid parthenogenesis. Now we have come across apogamy, which is a parthenogenesis. Parthenogenesis is a type of apogamy, and now we have development of a haploid egg from into an embryo. Means the haploid embryo and plant will be developed. Now this is known as a haploid, or this is called haploid parthenogenesis. Okay. Now what happens is in other plants, haploid embryo formation, okay, has also been noticed from male gametes. That has been observed too, and those haploid plants are usually sterile in nature. That has been seen. Now, in diploid parthenogenesis, what will happen? How will you differentiate haploid and diploid? So, in case of diploid parthenogenesis, what happens is the embryo sac gets formed or is usually formed from any sporophytic cell of ovule. From any sporophytic cell of the ovule. Okay, and then what happens is it forms from sporocytic cell of the ovule, but does not, that ovule does not undergo meiosis event. Okay, that is again apospory, no meiosis. You can call it again apospory. So think how things are related. The diploid parthenogenesis can be called as, you can say, apospory, and all the cells of the embryo sac will be, you can say, all the cells of the embryo sac will remain diploid in nature. Will remain diploid in nature. So this is all about your apospory, apogamy, and parthenogenesis, haploid, diploid. Okay, so be very careful while solving, especially the NEET questions. Board exam questions directly, the definition that I gave you, that will come maximally. But in NEET, tricky questions from this can arise. Okay, as I've taught you in detail. Now coming on to the parthenocarpy. Okay, from parthenogenesis to parthenocarpy. Now once we come to parthenocarpy, okay, so what we have is the fruit is normally formed by stimulus of fertilization, that we are all aware of. Okay, the fruit, you can say, is normally formed by you can say stimulus of fertilization. Now what happens is sometimes fruits may be formed or may get formed without the act of fertilization also. Sometimes fruit formation can occur or can take place without, you can say, the act of fertilization. Without the act of fertilization. Now what does that mean? This type of event where fruit formation occurs without the act of fertilization, this is known as parthenocarpy. This is known as parthenocarpy. Now, what could be the reasons behind parthenocarpy? If we talk about the reasons, okay. So the reasons behind parthenocarpy is, or parthenocarpy driving reasons, okay, parthenocarpy. The reasons are, if I say number one, first reason could be the stimulus of pollination. Stimulus of pollination. So fertilization never happened. Okay, so you can write in bracket without fertilization. So stimulus of pollination may be enough, may be sufficient to induce fruit formation. May be enough to induce, you can say, fruit formation. That is there. Next is again parthenocarpy could be induced by several hormones. Okay. We all know that there are various plant hormones. We have auxin, we have gibberellin, we have cytokinin, right? We have abscisic acid, we have also ethylene, which is a gas, right? So parthenocarpy can be induced by or can be artificially, you can say, induced by various types of gibberellins, okay, by hormones. Neat question is this one. Parthenocarpy can be induced by which hormone? It can be induced by gibberellins. Can be induced by hormones. Meaning that it can be artificially induced by, you can say, artificial induction of parthenocarpy, right, can occur very easily if we apply enough amount of gibberellins and also some amount of auxin. Okay, a combination of gibberellin, okay, gibberellins which are acidic in nature, gibberellin acids, GAs, and auxin. Auxin is again acid, right? Indole 3 acetic acid is auxin, IAA. So if we apply this, we can induce parthenocarpy. Very easy. Okay, this is again a good neat question for all of you. So that could be the correct way to induce, but sometimes the thing is parthenocarpy is the natural or artificially induced production of fruit without fertilization of ovules, you can say. So the fruit remains seedless. Okay, so the fruit remains seedless. Very important. The fruits remain seedless. One more characteristic that you should be remembering. Okay, so if you are talking about hormones, so when sprayed on flowers, any kind of the plant hormones, gibberellin, auxin, cytokinin, can often stimulate the development of parthenocarpic fruit. Even cytokinins have been found in research which can go for parthenocarpic development. That is artificial parthenocarpy. But the thing is, it is not required mostly because plant hormones are seldom used commercially to produce parthenocarpic food. Or automatically parthenocarpy occurs. So it is not required, okay, on commercial basis. Okay, so that doesn't need to be done, but it can be done. Okay, mostly gibberellin and auxin you will be putting the tick on, but cytokinin also has been used in some cases. Okay, along with gibberellin and auxin. Now, coming on to the next topic known as seeds. Okay, so a seed is a ripened fertilized ovule. A seed is what? It is a ripened and fertilized. That's why parthenocarpy, no seed is produced because no fertilization occurs. Fertilization does not happen for fruit formation. So a seed is a ripened fertilized ovule. Is a ripened and fertilized ovule, which is the final product of sexual reproduction. It is the eventual or final product of sexual reproduction. That is what a seed is. Now, what does it contain? It comprises of an, you can say, embryonic plant, reserve food, and the protective coat. Embryonic plant, there's a reserve food material, glyoxylate cycle, they can go on from fats, they can produce glucose, right? And protective coat. So we cannot the humans, I'll come to a little bit of biochemistry here. So the humans cannot achieve such kind of milestone, right? If you have studied Krebs cycle properly, so pyruvate is converted. Okay, you have COA, you have thymine pyrophosphate, you have lipoate. Okay, this is an oxidative process, so this will be reduced NAD plus. Okay, it will be NADH plus H plus. Okay, so what happens is one CO2 will come out, you will make acetyl coenzyme A in the Krebs cycle. Okay, just listen to this carefully. Now, the enzyme used is pyruvate dehydrogenase complex, which is a huge enzyme. It is made up of E1, E2, E3. Okay, E1 is pyruvate dehydrogenase. Okay, E2 is dihydrolipoyl transacetylase pyruvate dehydrogenase. This one is dihydrolipoyl transacetylase. This one is dihydrolipoyl dehydrogenase. Now this converts pyruvate to acetyl coenzyme A in the matrix of the mitochondria inside the inner membrane after the pyruvate has been taken in by the mitochondrial transporter protein. Okay, but this now acetyl CoA can react with, you can say, oxaloacetate. Okay, and citrate synthase will be the enzyme can form the first product citrate. Okay, or citric acid. Okay, and that is the product which has 3 carboxylic acid, so tricarboxylic acid cycle, or you can call it Krebs cycle, or you can call it citric acid cycle. But once acetyl CoA has been produced, or once acetyl CoA concentration goes too high, and if you need a lot of glucose in the blood, you cannot convert acetyl CoA into pyruvate. Pyruvate can be converted to glucose in the back reaction known as gluconeogenesis in human beings. It is the opposite of glycolysis. Pyruvate can be converted to glucose, but acetyl CoA cannot be converted to pyruvate in any mammal or in animals. Okay, but plants, the oil, the omega 6, omega 9 fatty acids, the oils that they contain in the seed, okay, if at all they have acetyl CoA, that can be converted back to pyruvate, okay, via the glyoxylate cycle. This happens in the glyoxysome, which is types of peroxisomes in plants, glyoxylate cycle. So that is a peculiar biochemical mechanism present only in the plants. It is devoid in case of, we are devoid of it. It is absent in case of animals. So this gives them the immense advantage of converting acetyl CoA to pyruvate. Okay, because acetyl CoA, once a fat is broken down, a fatty acid is broken down via fatty acid catabolism. Why it is so important? Because once a fatty acid catabolism happens, once a fat is broken down, okay, because fat has COH, so we call it fatty acid due to the presence of COH group. Okay, so the thing is, when it is broken down, what happens is, due to the catabolic process, the acetyl CoA molecules, they are released. Okay, and that acetyl CoA molecule, if they can be, they can be converted to, you can say, pyruvate. Pyruvate can then be converted to glucose via gluconeogenesis. Okay, to glucose, opposite of glycolysis. Now, what you can say? You can say that you are breaking fat and using fat to derive glucose from the fat, which animals cannot do. That thing can be directly achieved by the plants. Okay, so a new generation of plants commences with what? Commences with the formation of seed. Now we know that the embryo, which is also known as the kernel, okay, embryo, which is also known as the kernel, is the future plant in miniature condition. Is the future plant in miniature condition, you can say. Now, when the seed is sown in soil, a new plant appears from the embryo only. So, in seeds, the metabolic activities also get suspended. Okay, if there, you can say, circumstances are extremely unfavorable, unconducive, so they can suspend the metabolic activity. Okay, now the embryo in the seed is made up of the embryonal axis. The embryo in the seed, I'll show you the figure also. In the seed is made up of embryonal axis. Now, it does comprise of radicle. Okay, radicle is nothing but embryonic root, which will become root later on. Embryonic root. Then plumule, which is the embryonic shoot. Embryonic shoot. So I'll just show you the image, okay, of a seed. Seed anatomy. You can see this one. You have seed coat, you have plumule, you have hypocotyl radical axis, you have the hilum, you have the micropyle, you have the cotyledon. Okay, so we'll talk about all this here. So what happens is, on one side, one or two embryonic leaves or cotyledons are present, just like you can see here. Okay, in some seeds, the food is stored in the endosperm. Okay, now the seeds which store their food in endosperm are known as endospermic seeds or albuminous seeds. Okay, the seeds, that is very important, which store their food in the endosperm are known as endospermic seed or albuminous seed. For example, you can have the seeds like castor, bean, okay, wheat, rice, maize. They're all albuminous seeds. Okay. Now, the seeds which store their food in cotyledons, where the endosperms are absent, are known as the seeds which store their food in cotyledons. Okay, where the endosperm is absent. Endosperm is absent. Are known as, or are called, we can say, non-endospermic or X-albuminous. They were albuminous, is X-albuminous. X-albuminous. You have the example of Pisum sativum, Gregor Johann Mendel, pea, gram, groundnut, etc. Groundnut, etc. Now, if you see the structure of a, you can say, dicot seed, okay, so the, especially if you see the structure of bean seed or the structure of maize grain, okay, so those things I'll tell you. So let's start with the structure of a maize grain, okay. So maize grain structure, that is a monocot endospermic grain, okay. So maize grain structure, you'll be able to see it here, okay. You can look at it and I'll describe it to you, okay, from the image. So it is one-seeded fruit. You should know that it's a one-seeded fruit, okay, which is known as caryopsis. Okay, one-seeded fruit is known as caryopsis. So structure of maize grain. Okay. Now it is a one-seeded fruit known as caryopsis. Now, or grain, because the pericarp means the fruit wall, because pericarp slash fruit wall is fused with the testa. You can look at the image, it's fused with the testa. You can see here the pericarp is completely fused with the testa. In the image, you can directly see. And then you will be asking yourself, what is the main difference between a testa, and you might have heard of something else known as tegmen. Okay. So the main difference between a testa and a tegmen is that testa is the outer seed coat, whereas tegmen is the inner seed coat. Always remember. Okay. If you want to know more, testa is smooth, thick. And say testa is smooth, thick, impermeable. Okay. And may be colored while tegmen is a thin membranous structure made up of hyaline. Thin membranous structure made up of hyaline. So that is the tegmen and tesla. Okay. Now tesla and tegmen are the two seed coats which are found in the bitegmic seeds of dicots. But here, since it's a monocot, okay, it is fused with testa. Now you can see the seed coat here. Now the seed coat, it is the, in case of maize, I'll first write it down. When you look at the seed coat, okay, it is the outer brownish layer of the grain. Brownish layer of the grain, right? What happens is seed and fruit wall are completely fused together. Seed and fruit wall are fused together. can see here, seed and fruit wall are completely fused together here. Okay. Then we come to the endosperm. When we come to the endosperm, what you see is this is the longitudinal section LS. Okay. So it comprises the major part of the grain. Okay. So it comprises the major part of the grain and is filled with reserve food. Okay. It comprises of two regions you can see. You can see here the allulose layer, right? So the outer single layered allulose layer, which is mainly made up of alluron proteins, and the inner starchy layer made up of endosperm. Now it is separated from embryo by a layer of cells which are called as the, you can say, or which are known by the name as epithelium layer. Okay, that is how it is separated from the embryo. So you can see the endosperm. Okay, it contains a single, or it comprises of, you can see, it comprises the major part of the grain, and is filled with food reserve. Okay, and is two layered. Okay, two regions. Region number one, outer single layered alluron layer. Alluron layer made up of alluron proteins. Then inner starchy endosperm. Inner starchy endosperm. Okay, and it is, you can say, demarcated or separated from embryo by a layer of cells known as the epithelium layer. Now we talk about the embryo. Okay, I'll write it down and then I'll show you. When we talk about the embryo, okay, so embryo comprises of a single lateral cotyledon. It comprises of a single lateral cotyledon, and that is known as scutellum. Scutellum, very very important. Okay, and embryo axis is with the plumule and radicle which are at its two ends. Embryo axis does contain plumule and radicle which are at its two ends. Okay. Now the root cap would protect the tip of the radicle, and the radicle is surrounded by a protective sheath called coleorhiza. Okay. Radicle. The very important NEET questions are covered by an important sheath, okay, known as, you can say, protective important sheath. You can say protective sheath. Important is fine, but it should be protective here. Okay, by a protective sheath known as coleorhiza, right? Now plumule is also protected and covered by a sheath. Okay, plumule is also covered by a sheath that is known as coleoptile. coleoptile. Now you can look at this image. Okay, you can see the coleorhiza here. You can see the scutellum here. Okay, so coleoptile also you can see here. Okay, now you'll be able to see this embryo and look at the different markings that have been done, different, you can say, labeling process which has been done on this, right? So this is what the maize grain in its entirety is. Okay, that is very, very important for you to know. Okay, now if you compare it with the dicot seed like castor seed, okay, so if you just see the castor seed, I'll explain it to you. The castor seed, you can just compare. Castor seed diagram. Structure. You can see here, okay, you have bigger structure, castor. Okay, this is better. Okay, so you can see, you can just compare the two structures. Okay, so here what you have, it is broader at one end, right, and pointed at the other end with an elliptical outline, you can say. Now the color is a mottled dark brown color, okay, with hard, smooth and shining testa. Now the tegment is thin, white and membranous. Now it's also, what can be noticed, it's a soft white spongy structure, okay, which is known as, it, in this diagram it hasn't been put, you can say, known as caruncle, is also present at one end of the seed. Okay, it is nothing but an outgrowth of the micropyle, you can say. Now you have the raphe also present in the middle of the flat seed. A narrow raphe is also present. Now there are two cotyledons which are thin, oval, peppery and veined. Now the endosperm is very, very fleshy, in which the food is really stored, and embedded in the endosperm what you have is, you can say, the caruncle will be this one only. If you want to see, this one is the you can say caruncle. Okay, this is the caruncle, this one is the caruncle. Okay, so it has not been labeled, so I'm just telling you where the caruncle will be. So embedded in the endosperm, okay, will be present something like the embryo axis with distinct radicle and plumule. So that will be the case here. And epicotyle, hypocotyle that you are seeing here, okay, so I'll just tell you the difference between epicotyle and hypocotyle also. So epicotyle is which extends above the cotyledon. Epi, like epidural, extends of the dura mater of the brain. Epithalamus, over the thalamus. Okay, so it extends above the cotyledon. Okay, and is composed of the shoot apex and leaf primordia. And you can say, if you talk about the hypocotyle, which is the transition zone between the shoot and root and the radicle below the cotyledon. Okay, that is known as the hypocotyle. So this is the comparison between, you can say, a maize seed and a castor seed. Okay, so now the next thing is, we can talk about one more thing known as seeds as physiological enigma. So seeds have been considered, okay, as physiological enigma. Why? Okay, so I'll first write the definition. I think you know it already, so there's no need to write the definition of epicotyle and hypocotyle. So we can talk about as seeds as physiological enigma. Now, seeds have been considered as physiological enigma of the living world. Why? They contain a miniature plant a protective coat, reserve food for future growth, and also, okay, protective seed coat is fine. You can say reserve food for future growth, okay, acceptable, that is fine. Miniature plant, okay, we all know that. But it also contains, you can say, few long half life, or you can say long lived RNA molecules. These mRNA molecules, rRNA molecules, tRNA molecules are present. Okay, and these RNA molecules can guide future metabolic events and growth events too. Right, future metabolic events and growth events. We know that without mRNA, okay, can you form protein? You can never form a protein. And if you cannot form any protein, how can you form the enzymes? How can you go for metabolism? Metabolism nothing but a culmination and amalgamation of catabolism and anabolism. Both require a plethora of enzymes for the perpetuation of various biochemical cycles. So you talk of glycolysis, talk of gluconeogenesis. Then if you want to make glycogen, you have to go for glycogenesis. Then we have to go for glycogenolysis if you want to break down the glycogen. Same way fatty acid catabolism, fatty acid anabolism, nucleotide catabolism, nucleotide anabolism. Then cholesterol, you can say cholesterol synthesis, then synthesis of various hormones. Then coming on to the protein metabolism, amino acid synthesis, amino acid breakdown, those transamination reactions. So many reactions are there. Krebs cycle is there, electron transport chain is there. Upon the ATP, you can say ATP production happens in the inner mitochondrial membrane through the complex site known as F1, F0, ATPase. That is there. So there are more than 300 biochemical cycles which run parallel to each other. You have hexose, monophosphate, shunt pathways. So many are there, and those are important for the growth and development of the body. And without mRNA molecule, you cannot have the enzymes. Without enzymes, nothing can be catalyzed. They are the biocatalysts. They reduce the activation energy. This is the difference between the, you can say, ground state and the transition state, right? So if that is not lowered, the activation energy, how come the reactions will continue, right? You have the lock and key model. Also, the substrate is the key, the enzyme is the lock, right? So the thing is, it is really, really enthralling and interesting to note that living cell dies if water content falls below 45 to 75%. Okay, a living cell will die due to necrosis if water content falls below 45 to 75%. It falls below this range. But seeds continue to live with 10 to 15% water only. They continue to live with 10 to 15% water content. Okay, so we can say that in seeds the metabolism is suspended. Is suspended. Okay, just like you can say, you can compare it to the hibernation estivation of the animals. The hibernation is achieved by uncoupling protein, okay, uncoupling factor protein, okay, that transits the energy from, you can say, generation of phosphorylating the ADP. It doesn't want that, so it will deviate the phosphorylation of ADP event through the F1 F0 F1 F0 ATP synthesis in the mitochondria complex site to the formation of heat. Where this uncoupling factor protein is used in, you can say, polar bear. Same way, we have suspension of metabolic activity in case of seeds. So in seed, all of a sudden, growing embryo becomes dormant and it can stop its growth. That happens quickly, so quickly that it's a sudden event. Now food becomes immobile and respiration becomes anaerobic in mature seed. That also happens. Now seeds are also able to tolerate higher low temperatures. tolerate high and low temperatures. Now, what happens is, in the end, at the time of germination, the seeds do break down the chemical inhibitors. Okay, the chemical inhibitors means the inhibitors which stop or which prohibit the seed from secreting growth hormone, and at the time of germination, the chemical inhibitors are broken down, and ultimately the growth secreting, or you can say the growth hormones, they are commenced to be secreted. The seeds start secreting growth hormones. So growth hormones level, they rise ultimately in a seed. Okay, so this is all about the seed. Okay, in the next class we can finish this chapter, dormancy, apomixis, fruit, and we'll commence the next chapter also, human reproduction. Okay, then, so I hope you were able to comprehend the events. Okay, 5 minutes it will take to finish this chapter in the next class, 10 minutes only, not 5 minutes, 10 minutes, and then we can move on to the next chapter. So I hope you were able to comprehend the lecture today. And if you have any kind of doubts or queries, you can contact me on my personal number for my prompt reply. Okay, then, thanks a lot. Let's see you in the next class.